origins of life, origins of the universe, and other things like that, also origins of humanity. So I got really invested in that, and I was just really, really interested in the origins of things. And so I had Dr. Stone, who, if anyone's in bio at Mac, you may be familiar with him, but he's just the loveliest man in the world, and he encouraged me immensely. He took me on as an honors thesis student. So when you have to do an honors thesis, if you're in honors biology at Mac, that's one of our requirements. And so I got to work with him. And he also made me a teaching assistant in undergrad for undergrads. So that was like a really cool experience as well. And I got to work on tardigrades, which are these cute little guys that you see here in this figure. And they're colloquially known as water bears, which is just a fun name as well. So I had a lot of fun working with Dr. Stone, and also being able to do research and knowing that I like research. And so from there, I was doing some elective courses in anthropology, and I really, really like those too. And so I have Henry Schwartz here. He's a geology and anthropology professor, and Dr. Stone connected me with him. And so I did some research with him on scurvy, and just had a lot of fun times just chatting with this wonderful guy, an emeritus prof, so sort of semi-retired, just chatting with him about life. And so that was really, really great. And then he actually, during this time, I was also taking all these courses. So I was taking forensics, I was taking what can you learn from ancient skeletons, I was taking courses on Roman civilization, and just like eating that up. I just loved it. And so one of those professors was Tracy Prouse, who's pictured at the bottom here. And so I actually got to go, at end of undergrad, I got to go on a dig, her excavation in Italy. So digging up ancient Romans. So here is me, little young Jess in 2011, digging up a skeleton with some of the other people on the dig. And it was just a really wonderful experience. And so I also have another image that's a little bit closer up. So this is a different skeleton. This is the knee area. And so this would be your like thigh bones and your shins. And so we have some cool stuff buried here. So we've got a nice lamp, a pot, another pot, and some nails. So it's just like the best experience ever. And I was like, yeah, this is for me. And so from there, Dr. Stone and Henry Schwartz wrote me really lovely letters of recommendation to get into my master's and later my PhD. So went to U of T for my PhD. And I just wanted to note that I also had a little bit of a failure issue here. And so I wanted to talk a bit about failure. So whether it be failure or just having a hard time. So when I started at Mac, as I mentioned, I had a really hard time. And I like to mention this because I think it's important to normalize that struggle happens, and it happens to lots of people. And I think that it should be talked about more. And so I got to my new program here, and I really enjoyed the work, but it wasn't something I was super passionate about. And I also wanted to mention that I applied to Mac as well for my master's to work with Tracy, but my idea I had proposed was really general. It was like, I want to study disease in the past. And so I was not accepted. I think they thought like, you haven't really honed your plan here. And so I wanted to mention that too, that I actually failed at getting into a master's at McMaster. And a lot of the people that I know failed first try getting into a grad program or getting into one of the grad programs they were interested in. So just another thing to mention in case, you know, this is useful for you or useful for you in the future maybe as well. And so I studied human skin pigmentation and looking at genetics in modern humans to understand skin pigmentation in the past and the evolution of it. And as I said, I liked the work, but I didn't love it. And so this is me in my lab group and my master's supervisor, Esteban Pera, on the left there. But I definitely wanted to go back to working on ancient human remains, getting to excavate and working on skeletons. And so that brought me to infectious disease and getting into my PhD at Mac. And so I now work on infectious disease in ancient Romans and medieval Romans with Hendrik Poinar and Tracy Prouse. And so Hendrik Poinar and I are here in this photo that I just love. It's us at the Ontario Science Centre pretending to be scared of a cave bear. And then Tracy you saw in the previous photo. And so my day-to-day involves things like lab work. So we have a sample here with bone that has been turned into a nice fine powder, and then it'll go through the process of getting ancient DNA extracted out of it. And then I also have, I know this picture is not great, but I also do a lot of computer code. So this is me and my coworker in my office, which is also my living room. right now. And so I do a lot of that as well. And I also do a lot of writing. And I also get to teach as a teaching assistant, which I really enjoy as well. Okay, so why did I want to do infectious disease? Specifically, I wanted to do it in ancient Rome. So ancient Rome covers this whole map that you're seeing here. And I was really wanting to look at Italy and see what kind of diseases people had. And so the idea being that we think there was a lot of disease happening at the time. So this time period that you're seeing here, so the map with the red highlighted showing that this is all of ancient Rome, we are at 117 Common Era, or you could also call that AD, so after zero, essentially. This is when Rome is at its height or its prime, and so you can see part of the Roman Empire is England and things like that. And it goes all the way down to North Africa. And so we know there was a lot of movement happening of people and movement of goods. We also know that populations were increasing immensely. And in addition, we know that there were larger groups of people in cities, so like crowding in cities and density in cities as well. So all of those things can lead to a lot of infectious disease kind of rising, as you can probably imagine. And so weirdly, we don't know that much about infectious disease in ancient Italy specifically. We have a bit of a better idea in England, but in Roman Italy, we're not very, we just haven't done enough research. So my plan was to come here and study infectious disease in ancient Rome. And so I've looked for things like gonorrhea, syphilis, brucellosis, smallpox, leprosy, TB, I think, are the main ones that I've looked for in ancient human remains. And so I've looked in remains from places like southeastern Italy on that dig site that I showed you guys pictures of. I also have looked in medieval Italy as well, so not quite Roman, but we'll talk about that later. I've looked at one site in Turkey and also Pompeii kind of area, and we'll talk about that more as well. So near Mount Vesuvius. But so that's sort of what I came in planning to do for my PhD. Okay. And so, oh yeah, one other thing I wanted to tell everyone was, so we thought that diseases like tuberculosis and smallpox were probably around during this time, and they're probably endemic, right? So you just have tuberculosis around and you have malaria around. And maybe there's sometimes like flare-ups and things like that. But we also know there were really big epidemics at the time, and so we could call those plagues as well. So we have things like the Antonine Plague, which happened in 165 Common Era. It might have been smallpox or measles. And we think the mortality was around 7.5 million people at the time, but it's hard to estimate these numbers based on historical documents. So 7.5-ish, but it could be more, it could be less. And then we have the plague of Cyprian, and that's another really interesting one. So that's, now we're moving forward in time, we're at 249 Common Era. And this is the bubonic plague, or typhus, or cholera, or smallpox, or measles, or anthrax. So we don't know what it was, but it killed a lot of people. And the quote here is about 5,000 a day. I think that was actually only in one particular town. So also killing off a lot of people. Then we have the plague of Justinian, which is very, very interesting as well. So the Emperor Justinian got this one and didn't die of it. So that's just a fun fact as well. And so this one, we think that it might have come from Italy based on historic documents, or sorry, not Italy, Egypt to Italy based on historic documents. And that it might have been influenza or bubonic plague. And at least that was the thought for a very long time. And then in 2014, a gentleman from our lab actually found Yersinia pestis, which is the causative agent of bubonic plague, in a sample from around that time period. So we now know that the Justinian plague was caused by the bubonic plague, which is again Yersinia pestis, which also causes the Black Death. So I know there's a lot of words for this sort of disease and how it takes place in different time periods. So I hope that's clear. Okay, so that was my big slide on Italy. So I just thought I'd ask a really quick question here is, what do we know about disease in the past? So if you guys or you wonderful humans heard anything about disease in the past, like a cool diagnosis or something you've read in the news, or maybe you have a friend that's taking anthropology and is really telling you stories or something like I would have been doing. So feel free to pop it in the chat. I don't think you can verbalize right now, but chat would be groovy. And there's no wrong answers. This question is very open-ended. and thinking maybe nobody. Oh, we
No,
got
wait,
one.
sorry. There, we're getting some answers, but there's just something off with the settings. I'm
Oh,
so sorry.
something wrong.
1
Okay.
second.
I see.
Yeah.
Okay, thanks for letting me know. I see Spanish flu, so that's good. I'll take the one that I
Yes,
that I
there we
can
go.
see. No, not at all.
Um,
Oh, I also see, for what I remember, a lot of disease was connected to religion. I think I see too.
yeah, like those, and then some other answers. Let's see. I'll just read through it because it was like direct
Yeah,
messages
that's
to me.
great. Thank you.
Someone said Spanish flu, and then someone said lots of people would die from it.
Mhm,
And then someone else said that there's no proper treatments and that's what would happen. And
mhm.
then yeah, the note, that's all. There's like the ones that are hidden from this.
Yeah,
It was like
yeah.
directly messaged to me. I don't know what happened.
Oh, I see. Yeah, thank you for reading those. Yeah, and I see
No worries.
disease was well understood, and there were lots of theories about what caused disease, like humors. Yeah, so these are all wonderful answers. So we got herbal medicine, the Black Death. Yeah, beautiful answers. So there's tons of different fun things we know about disease, right? And yeah, it could be bloodletting to try to solve disease. It could be anything like that. And so I just wanted to know sort of where we were at. And all of these are fantastic answers. So, and unfortunately, I don't have time to elaborate on them, but I appreciate all of those answers. Okay, and so some of the things that were mentioned in the chat, I would kind of think if we're talking about learning about infectious disease in the class, in past, sorry, it's going to cover some different categories. So typically we look for things like presence. So yeah, can you find Yersinia pestis in a Black Death individual, someone who died of the Black Death, or the Justinian plague that we just talked about? Geographic and temporal spread, so where was disease, that kind of thing. How did it move? What time periods was it in? We have evolution, so how did the disease itself change? So specifically referring to like the actual bacterium or the causative agent of the disease. We have things like transmission and risk. We have impact on humans, and so that could be biological, but it could also be social. So it could be something like, I don't know how familiar everyone is with the Black Death, but it killed off about a third of the population of Europe. And so after so many people had died, people like farm workers that didn't make a lot of money before were able to say, like, if you want me to, tend your crops, I want a better wage because I can go next door and I can help the next guy because there's not many people around to help you. And so this actually changed the economy of the time. So it was sort of a supply and demand thing. So, and there's lots of other cool Black Death things we could talk about. And again, Black Death is medieval, so I'm being a little sneaky here. I'm talking about medieval stuff during a Roman talk, but it's just a really good example. And then we have how the past can inform the present. So it could be things like learning about disease reservoirs. It could be learning about things like the spread of disease and risk of disease, and maybe how humans contribute to things like that. And then, of course, probably one that you might all be thinking about is stigma and blame. So we see that in the past, and we see that now with COVID. You see it, unraveling or unveiling itself in front of you. Okay, so how do we study infectious disease in the past? And so when I say we, I mean anthropologists, but I also mean other people that study disease too, because there's historians and other cool folks that are also interested in these things. So we're going to use the Black Death as an example here, because again, it's sort of, people have at least heard of it. And so I usually find it's a good kind of way to get into these things. And so again, as I mentioned, a sort of a primer to the Black Death, it killed about 30% of the population of Europe. And this was from about the 1300s to the 1800s. It sort of came and went in waves. So it'd be like, it's killing a lot of people and then it kind of goes away for 20 years and it's killing a lot of people and then it kind of subsides. So that's sort of the idea that we're going with. And so we have things like the historical record. So we have historians, or sorry, I shouldn't say historians, people writing stuff down at the time. So Black Death is going on and somebody's writing notes saying, like, we think it came from here. And so we, based on historical information that people have read, we think that the Black Death actually kind of began in Kaffa, which is in modern day Crimea, which is Russia or Ukraine area. And we think what happened was Mongols, so nomadic people that are often on horses, they were fighting with Italians in the town of Kaffa. And this was an important port city. So they were fighting about like who gets to trade stuff. And they had apparently brought disease with them. So everyone's fighting in this battle and the Italians are inside city walls and the Mongols are on the outside. And the Mongols, whether knowingly or not, started, like, they catapulted their dead over the walls. So I don't know if they knew that they could possibly be giving people disease, because obviously they didn't understand germ theory like we talked about. They were probably more in like the humors kind of idea. But either way, it was basically the first, we think maybe the first bioweapon kind of thing. So they were chucking their dead over the walls of the city, and the Italians got really sick. And so from there, the Italians fled when they were able to, and we think that that sort of Black Death was able to move from there, and it then started spreading via different trade routes. And so this is sort of the map of the Black Death. So that's the sort of historical narrative we have for transmission and spread of the Black Death. And then we have things like art. So we have this picture from 1600s London, England, and you can see the sort of fear and chaos that's happening. You can really visualize it in this image. So you have essentially death dancing on the graves of people. And you also notice the people are actually unburied. So it's not their graves, sorry, it's their coffins. And it's telling you to keep out of London essentially too. So you can see the essentially terror that was happening at the time. And then we have archaeology. So also another cool one. And so archaeology, we can do things like find mass graves. And so often if you find, you know, you're building a parking lot and you find a bunch of skeletons, if it's a mass grave where people are kind of piled together, it usually means that people died really quickly and then they were buried quickly. And so that could either mean usually infectious disease, or it might mean that there was like a battle or something like that. And so in the case of the Black Death, we actually have documentation. So going back to tying the historical record to archaeology, we have documentation of specific plague burials of like, we buried our dead here. So this is East Smithfield, which is a site in London, England, that this is a known plague burial. So just really cool. So our last two that I'll cover today are zooarchaeology. So interestingly, and you may not know this, and that's totally fine if you don't, but plague, typically bubonic plague, is transmitted from, there's little rats and there's little fleas on the rats, and then the fleas bite humans, and then humans get plague. And so there are areas where we don't have good archaeological evidence of rats being present. So places like Norway, they haven't found a lot of rat bones. And so people are trying to figure out how could it be being transmitted if the main host of the Black Death or the plague is not there. And so that's why I have this cute little cartoon with the mouse being like, wasn't even in Norway, sorry, the rat. And so we'll cycle back to that later too. And then our last thing, which is more so what I do, although I also try to do all these other things, whether it's poorly or not, is for someone to judge, I don't know. But I also do paleopathology. And so that's looking at human remains to understand disease in the past. And so that could be just looking at a bone and trying to see if you think it has disease, because some diseases actually manifest on bone. Or it could be for something like the Black Death in this example, it kills too quickly to impact your bone. And so what we do is look for things like DNA or protein in that bone. And so we actually have Jen here, who's a past Pointer lab member. So that's the lab that I'm part of, with a little sample here that she's going to process and try to find Yersinia pestis in. Okay, so before I get a little bit more into more specific me, and then a little bit more about DNA, and then a bit of interactive workshop, I just thought I would point out, since this is a Women in Science workshop, all of the wonderful women in my lab, past and present, that have done really awesome work. So we kind of circle back to those questions that we talked about, that we can answer about infectious disease, using all of those lines of evidence that we had, but including DNA, which is what most of us do, and then try to pair in some historical things and stuff like that. So we have Marie-Helene, and she studies smallpox in 1800s Quebec. So she's doing presence kind of work. Then we have Sam, she does geographic and temporal spread and origins, or sorry, that's not right. She does not do that. Sam does the presence of oral microbiomes. So she's trying to figure out what people's oral microbiomes were like in Denmark during around the time of the Black Death. Then we have Catherine and Ravneet, and they do geographic and temporal spread and origins, and they work on the Black Death in Europe, but also in Asia a bit. And then we have the lovely Anna Duggan. She's a former postdoctoral student, so she's no longer officially physically in the lab, but she still works on research with us. She does smallpox research, so a lot of evolution of smallpox, if you saw maybe that paper or that headline come out a few years ago. And then we have Maddie Tapson, and she does transmission and risk of the Black Death. And her work is actually seeing if there might have been other rodents present in Norway at the time of the Black Death. So she's thinking like, well, you didn't find rat bones, but could there be other different kinds of rodents like marmots, which are actually a natural host of Yersinia pestis? that could be around that could have been responsible for getting close enough to humans for fleas to hop onto humans. So really, really cool work. And then we have Steph Marciniak, also a former PhD student, and she works on malaria. So she's studying transmission and risk of malaria in ancient Rome. And her work, she tries to find the DNA, so the presence thing that we talked about, but then she's also incorporating things like social and political and epidemiological aspects that might lead to increased risk of disease. And then finally, we have Jen, who was in that picture holding up the tube, and she does how you could be impacted. So humans are impacted by disease, and she does it in lots of ways, but one of the ways is actually biologically how you might be impacted. So maybe there was a change in immune genes after the Black Death in the surviving people that were left. And then all of us try to tackle how we can inform modern disease as well. And also definitely there's overlap in the questions that we're answering as well. So that's my awesome women in science thing. So there's a lot of us. And I actually, weirdly, all of the women in the lab study disease and all of the men study like mammoths. I don't know why that is, but so yeah, it's all women who do disease in my lab, which is really great. So now that I've told you all of this other stuff, what do I really do? So I now study brucellosis. And so that's a zoonotic disease. And I'm looking for it again in ancient Rome, but also in medieval Italy. And so if you haven't heard of brucellosis, that's okay, nobody has. So it's totally fine. So it's a disease, it's zoonotic. You can get it from animals. So I have all of the cute animals that you can get it from. They're adorable. So from hanging out near them and aerosolized bacteria getting on you. You can also get it from unpasteurized cheese. So that's fun too. And so it's significant to public health because it causes fever, muscle pain, and depression, and also fatigue. And so it can actually really wipe out like a whole workforce if people had it. And it's still very common in not in North America, but in like different parts of Africa, in the Mediterranean especially today, parts of Asia and parts of South America as well. And it's very interesting in the past because we don't know a lot about how present it was. Which is weird because brucellosis is actually a disease that shows up on bone. So if my picture comes up, hopefully it does, we'll do one more click. Yeah. This is an example of an archaeological find. So one of the few finds that we have for brucellosis in the past. This is from Betrun, Albania. So it's medieval period again, not quite Roman. It's the 12th century. And so these holes in the spine, those are not good. That's not normal. If you haven't seen vertebrae before, that's totally fair, but these guys shouldn't be there. And so that's a brucellosis sort of indicator. Tuberculosis can also look similar, but we're pretty sure that this is probably brucellosis. And so the question is, if it shows up on bone, and actually it shows up on bone in up to 80% of people that have it, why don't we have more skeletons with these cool holes? And so what I'm working on is thinking about all the reasons that it may or may not be there, but specifically also trying to find it with ancient DNA, since for some reason we're not finding it on bone in the past. Oops. Try that again. Sorry, everybody. Okay. So I was worried that I would not find brucellosis in the past because ancient DNA can be really fickle. We're working on really old samples with really degraded DNA. And so often you go in with a project and you're not going to find anything. So I got really lucky. We had this wonderful guy, Santi Brancarsini. So he's dead when I'm calling him this wonderful guy. He's an Italian saint from northeastern Italy. And so this is him here. He was buried in a crypt in the 1300s in medieval northeastern Italy. And so he actually was buried in this, sorry, I say crypt, but altar is probably more appropriate. He was buried in this altar here. And he was excavated in the 1990s. And recently, about 3 years ago, he had these kidney stones that we were sent, and they were sent to our lab, and they were sent to us so that we could look for possibly tuberculosis, the bacterium responsible for TB in them, because tuberculosis can cause kidney stones, which is what these are presumed to be. And so weirdly, we found not tuberculosis, we found a ton of brucellosis. And so the bacterium that causes brucellosis is called Brucella melitensis. And so that's what we found in the sample. And so I was really lucky because that meant that I could keep working on brucellosis. And we'll talk more about that later. Oh yeah, and I also wanted to mention, so this is his pelvis, Brancarsini's pelvis. There wasn't a lot of evidence. I don't have a picture of his spine, which is really inconvenient because, as I showed you, you'd want to look for those holes in the spine. But the guy who got to look this over told me there's no cool holes in the spine. And so we do see a little bit of inflammation. So those are the little kind of spicules of bone that almost look like grass, if grass had hardened into bone here. That's evidence of inflammation, but that's not really very diagnostic for disease. It could be brucellosis causing it, it could be another infectious disease, or it could be something like trauma as well. So we wouldn't have found brucellosis in him without ancient DNA. So very cool, and ancient DNA is awesome. So continuing on, just thought I'd give you sort of a how we sort of conceptualize ancient DNA, and then how we do it, and then a little bit of workshopping. So we have our composition of our human before they die, and our lovely human here has all these different cells in them. So they have things like their own DNA, they have the microbiome, so maybe more like normal DNA hanging around, and then they might have pathogens. And from there, what will happen is you pass away, and then you have those green arrows that are now disappearing a little quicker than I would like them to, but you have those green arrows that happen. And that is essentially your endogenous DNA, so your original DNA degrading out of the sample and fragmenting. And so there's this sort of degradation and loss. Then we also have the orange representation here, the orange arrows, is contaminant DNA. So it might be things like the soil that the person is buried in that's coming in. And contaminant DNA is problematic because it can look like pathogens to us and be a little bit confusing. So it can kind of mismatch to things. And so that's why we are worried about the orange arrows, essentially. And so then we get our beautiful skeleton here, and we basically are trying to go back to the beginning. And sorry, pressing the wrong arrow. Trying to get back to A as best we can. And so that can be really hard because often the original DNA in the sample, so the person's own DNA, the bacteria, their microbiome, is actually less than 1% of what's in the sample, and that other 99 is like contamination. So, and then the pathogen that might be there is 1% of that. So it's a needle in a haystack. So very quickly here, I'll just show you how we do lab work. So we would get a tooth or a bone. We would then subsample that tooth or bone. So we have rab meat here, subsampling something for, I think, Black Death research. Then from there, we have our same kind of orange contaminant DNA that's in large amounts in that sample. And we're trying to pull the DNA away from other things in the sample, like proteins and minerals that kind of inhibit us from getting it out. And then we get it to the computer. And so we have our sequences, our nucleotide sequences of our like ATCG for our contaminants and our endogenous. And we don't really know what's contaminant and endogenous either. So I'm making this look really easy. Like I wish it was like some of it's orange and some of it's green, but that's something you kind of have to parse out along the way. So to figure out what's in your sample, which is sort of what we're going to be talking about today, is we use these different programs to try to classify all of those molecules. So all of those little chunks that we saw on the computer, and you'll have anywhere from 2 million to 30 million molecules typically in your sample. So it's a lot of classifying that happens. But how we go about it is we use programs like Kraken and BLAST that are databases of known organisms. And so then we try to match the DNA in our sample to a known organism. And so this diagram isn't totally accurate because it doesn't have pathogens in it, but the idea of how this works is the same. So it would say like, okay, for molecule 1, I know that it's probably a bear or a zebra, but I can't parse between them. And then I don't think it's a frog. And so then it would spit back at you. I can give you the class of the organism, and I'm going to tell you it's probably a mammal. And then you'll have other molecules that it's going to say, okay, I'm pretty darn sure that this is a black bear. And so you're getting a species level designation. And so same thing goes for the pathogens in the sample. It might say like, this is some kind of really broad pathogen and I can't really nail it down. Or it might say, I'm really sure this is brucellosis. And a lot of the molecules in your sample will remain either undiagnosed essentially. So it'll just say, I don't know what it is, especially for ancient DNA because everything's so degraded. Or it'll be something really high up, like it's an animal. And you'll be like, that's not very helpful. So again, just sort of showing how, I don't know, challenging this is. It's fun, but it's challenging. And so that process will be repeated for all of the molecules in your sample. Okay, so I'm going to give, I think, 2 examples if I can be fast, and then I will have 2 examples that you will get to go through, and we might have to switch it to 1, but that's okay. We can make it work. So I have a tooth here, or a tooth from a Plantis that this is near Pompeii. So if you're familiar with Pompeii, it was encased in ash after Mount Vesuvius erupted. And we've got lots of cool people from Pompeii that are like encased in ash, sort of frozen in time. And so Plantis is sort of the sister city, or maybe cousin city to Pompeii, and it's well known. But the interesting thing is that there's actually evidence that people find explosives holes at Pompeii. We don't have access to Pompeii for I don't really know what reasons, maybe political, but we have access to a Pontus. So we thought maybe there'd be brucellosis there if there was brucellosis nearby. And so this is what the output of the program Kraken that we use looks like. So when we're kind of diagnosing all those molecules in the sample. So you can see 24% that are in gray here are like, we don't know what it is. It's an organism. And then you'll see there's a lot of bacteria. And then down here we have Brucella at 2%. So that's the organism that's responsible for brucellosis. So this is one where I'm like, that might be real, and I'm still kind of working on that. So if you see a publication sometime in the future, then I guess I figured it out. But I just wanted to mention that. And there's no human in this sample, not because there isn't any human there, but because the database that I compared this to, Kraken, didn't have human in it at the time that I ran this. So it can't find a match if it doesn't have the thing in the database. So there's probably human in the sample, it just doesn't show as such. Then we'll do Brancorcini really quickly. So this is that kidney stone that you saw already. And then we have 10% Brucella, which is insane. Usually the numbers are not this high as we talked about, right? Usually your endogenous DNA for your whole sample is less than 1%. So this is 10%. This is really well preserved, probably because it's like a kidney stone and it's really hard. And then you can see in this case, I do have human in the database. And so we have 30% human. And most of that is original human, not contamination. So very, very cool. Okay, so I think for time, we'll maybe just do this one example, but I wanted to show you, so it's going to be your turn now, and we're going to talk about if you were somebody who was basically looking at DNA in samples, you're kind of trying to study nomad people and see what their ancestry is like, you end up finding Yersinia pestis in one of these samples. And this individual is from 180 Common Era, or AD, in China, from the Chen Shen Mountains, so up here. And you're very excited because Yersinia pestis comes a few hundred years later, so you're kind of wondering if they're related. And so what I'm hoping you'll do, we're going to put a link in the chat. And so it'll be the link that says pre-Justinian for my wonderful co-hosts, because I think you have 2. And I'm going to ask you if you'll click on the link and then you'll click on the pie chart itself and you'll see how many molecules in there are Yersinia pestis. And then you'll also look at what the percent of the total reads was. So what's the percent of the whole? Hopefully you can see that. And then one extra question is, why is Clostridium there? And you can Google Clostridium because you might not know, and that's totally fair. I didn't know before I started this research. Okay, beautiful. Yeah, so if people don't mind clicking that link, and I'll give you a couple of minutes to go through that. And so just to reiterate, if you're like, what is happening? You would click the link and then you can click on bacteria because Yersinia pestis is a bacteria. Then you would click on proteobacteria and then you should see Yersinia pestis. If you want to cheat, which is totally fine, you can also search Yersinia pestis, and I'll type it out here. In the top left box, there's like a search box. And then on the right-hand side of the pie chart, you should see numbers. So you should see near the top, it'll say like, I think total count, and that would be how many Yersinia pestis reads. And then if you go down to the bottom, there's like a couple of tiny, tiny pies. You should see the percent. So I'm hoping that that's fairly clear for people. Is it working for people if they're clicking on it? I hope so. Fingers crossed. Okay, I got some nods. That's good. Okay. Do we sort of have an idea? Do we need more time? What do my co-hosts think? Have we had enough time to look? Yeah, okay. I
I think that's
don't
good.
want to rush anyone. Great. Okay, so this is me honed into Yersinia pestis here. So I'm sorry that it's so terrible looking, but luckily you guys have your own awesome pie charts there. Does anyone know how much was in, like what the total count was for Yersinia pestis in the sample from looking at the chart? And feel free to throw it in the chat. Beautiful, yes. Excellent work, Christina. I'm pretty sure I TA'd Christina in the past too. I'm trying to remember. I think that's the case. Hi. Yeah, so perfect. Well answered. So yeah, it's 2000. And so yeah, you can see that here. If you were like, what is happening? It's up here on the top right. And then you can also see the percent of the total. And this one's a little bit harder. So if you don't get it, it's fine. But please do try to throw it in the chat. And if you're wrong, it's totally fine. So what's the percent of everything that is Yersinia pestis? And yay, I did TA Christina, yay. Anybody? 11% is a good guess. So that's a really good guess. It's not correct, but it's really close. It's close because pseudotuberculosis is actually the soil bacteria that's related to Yersinia pestis. So people are, yeah. So it's 0.02%, but the fact that Tanisha thought maybe 11 is totally understandable because it says Yersinia pseudotuberculosis complex, right? So totally fair. Yeah, so this one's a little bit harder. So I think typically if you're, so I do look at these charts all the time. And so it's sometimes weird when you do this all the time and you're like, how do I explain this to somebody for the first time? But it's that bottom circle there. So the root is like everything. And I don't know why it's called the root. I guess it makes sense. It's like the beginning of all the organisms or something. But yeah, so this is only 0.02% of the entire sample. So as you can see, it's like some needle in the haystack kind of stuff that's happening here. Okay, and of course I'm overly chatty, so we'll move on. Classic. But you can also see if you click yourself, and you can also keep that up and look later too. But we can also see common reagent contaminants. So we have lists of like people that are doing modern DNA, people doing ancient DNA, of things that they often see in their samples that shouldn't be there in like known samples. So we have things like actinobacteria and staphylococcus, and those ones, so I sort of know that those are contaminants, so I don't trust them if I see them. But in this case, it wouldn't be that this is mismatching and looking like a pathogen. Staphylococcus is a pathogen. It's just that it's not coming from the dead person, right? It's coming from the chemicals that we use to do the research that we're doing. And so you might, if you're inexperienced, look at this and go, oh my God, this person had a really cool staph infection. In reality, probably not. So I just thought I'd point that out because again, it kind of shows you that this is like a complex but very cool thing. And then we also have those soil contaminants. And I'm sorry, I feel like we don't have time for the question, but Clostridium is a soil bacteria. And so because this person was buried in soil, you get these bacteria that are common to soil as well. So that's why that's showing up. And if you got that, then big pat on the back, because I don't think I would have figured this out before I started this research. So. And then I think I have one more. So you'll see things like Neisseria, I hope I'm saying that right, that will show up in the sample as well. And that's actually an often oral microbiome species, but so Neisseria and then something else like Gondi, I think, Gondii, is a normal oral microbiome thing. So again, this is a tooth that we're sampling, so we'll see things that are common in people's teeth. But I myself have been like, it's been misclassified as Neisseria gonorrhea in my samples. So that's the thing I was talking about before where it can get confusing. And so I thought someone had gonorrhea in ancient Italy, and I was really excited that I found an STI in ancient Italy with DNA. But it was probably just someone's oral microbiome, and it was just mismatching a bit with the program that we use. So I think we'll skip this because I talk too much. I'm very sorry, but we'll continue on. But it's not that important. It's basically that you find out that your strain. Actually, I can say it really fast. You find out that your strain that you're working on, so the one that you guys looked at in this pink arrow, is really closely related to the plague of Justinian. So closely related being the vertical distance here. So they are really, really close, and that's really cool. And based on where your sample is from and other samples that other people have found ancient DNA in, you actually figure out that it probably was coming from ancient China, not ancient Egypt, as people had thought, historians had thought in the past. And so probably along the Silk Road. So probably the terrestrial version of that getting to Europe. So you're like, that's really cool. And I don't think we have time for the second one, but you guys have the pie chart, so you can always play with that in your spare time. And I can give my lovely co-hosts a bit of this information so that you kind of have some context so that you could play with this later. Sorry, I'm such a chatty Cathy. It's hard not to be a chatty Cathy. But what you've found essentially from doing this awesome work after looking in your pie chart that you can do later is you have found that the virulence factors that are associated with the Black Death, so this is a Black Death example, they are very, very similar to modern Black Death or modern plague or modern bubonic plague, whatever you want to call it. So the Yersinia pestis of back then and the Yersinia pestis of now are really, really similar. And so we've skipped to the Black Death, so I apologize, because before we were talking about the plague of Justinian. But you basically figure out that it's not really more virulent and killing people off in such large amounts and transmitting so well because of a genetic component of the bacteria during the time of the Black Death. It's probably things like socioeconomic factors, hygiene, maybe co-infection. So it's probably more social, political, epidemiological causes that made it such a crazy disease as compared to it being some weird mutation that made it so much worse. So also very cool. Okay, I got 2 slides left and I'm trying to be fast so we can have questions. I will just sum this up really quickly. So my point here, and we could talk about it more later, but my point here is that there was a lot of othering that happened during the Black Death back in the day. And we'll just skip to my third point. So we know there was a lot of blame on Jewish people. They actually thought that Jewish people were poisoning wells at the time because Jewish people were a minority and people disliked them. And so that was sort of a thread or a narrative that Jewish people were trying to wipe out Christianity by spreading this plague. And so now we see the same thing happening in other diseases, right? HIV, AIDS, SARS, and then obviously, as you all know, COVID-19. So we see this blame and we see this othering and we see conspiracy theories like it's a bioweapon from China. And so I think something that we can contribute to is knowing that this is something people do out of fear. Is also, something that happens because of discrimination that's been long embedded. And so we can try to provide that context and then hopefully have an idea about it and hopefully maybe prevent it in the future, or at least talk about it as a narrative that keeps coming and happening again and again. And then I have my in sum, but basically women do awesome science, failure and struggle are normal, ancient DNA does awesome stuff, and maybe we can help keep history from repeating itself. Thank you, and sorry for my chattiness.
Good.
So,
that was amazing.
Thank you. Yeah, so questions if everyone's up for it.
Yeah, everyone, you can just type in your questions to the chat, if that's okay.
And literally anything is fine too. It doesn't have to be like super specific DNA question or anything. Thanks, Christina. Okay, so she says, out of curiosity, how do you think that plagues in the past compare to our current ongoing pandemic, and is any of the research similar? Oh yeah, that's a great question. So plagues in the past comparing to our current pandemic. So I think lots of things are similar. So we now know lots of the same risk factors happen, right? So things like globalization. So in ancient Rome, as we had increased population size, trying to feed more people moving around, we we assume there's an increase in disease, and we see it documented historically, and we're learning more about that, as well with these other plagues and things and the DNA that we find. Then we have, are things like blame and stigma that we see just recurring and re-happening. There's probably tons of other things, but I can't think of anything right now. But I would say globalization and then like blame and stigma would probably be good ones. And how humans are an agent in this, right? Like we are the ones doing like a lot of farming or things like that. And that's happened in the past too. And so just remembering that, or we maybe make marshes that then have malaria be more common, like happened in ancient Rome, due to like siphoning water to certain areas. So that we need to remember that we can help with this too, and we can also hinder it. And I think a lot of the research is similar. Again, I can't think of a good like example off the top of my head, but I think a lot of the narratives are similar, and a lot of the like, you need to look at epidemiology, you need to look at social and political factors, and as well as just like basic biology and things like that. But it's really important to look at things like inequality and stuff. And I think people are realizing that for the past and the present too. The plague of Athens. I don't think we know. So that one is in Greece, as Rupali is mentioning. I think there's an ancient DNA paper which is not well received, where they think it was, it's some kind of like potentially soil dwelling bacteria. And so the fact that people found it and didn't do a good job authenticating it, it's probably soil contamination. And so I'm trying to think, it might be that they think it was typhoid, Salmonella typhi that they found. And so we still don't know, but if you Google it, you'll find that paper and you might be like, oh, and we don't believe that paper as of yet. How are we doing for time? We have 4 minutes. Is that right?
Yes, or 3 now. Okay.
Great. Okay. What else do we have? Oh my gosh, so many great questions. Do you guys want to pick one, co-hosts? I don't know what to choose.
Let's see. Okay, let's just like do like what is it? Okay, let's do Madeline's question. It just says like, do you ever observe mutations in the genome that have happened with time when comparing ancient samples with current data?
Oh yeah, for sure. So often you'll look in the past and you'll see, so you'll have the past and then you'll have your present one, and you'll have a lot more mutations because there's been more time to accrue mutations, if that makes sense. And so you'll see like differences and stuff. And we use that to build like the trees, which I very briefly showed you. So those trees, that branch lengths that you saw, so maybe you noticed, people might have noticed the Justinian plague, it had a really long branch length. So the horizontal line was huge. That's because it's got a lot of mutations. And that's probably because it was like rampant, sort of like COVID right now, where it's like moving around and moving through people and getting to reproduce. I hope that was a good answer.
That was amazing. Okay. Thank you so much. Again, I'm so sorry that we had to cut up the Q&A session like really short. Yeah, it's just like we're kind of like on the next schedule is like 2:10, but yeah. Anyways, that's all we have for this workshop. Thank you again, Jessica, for helping us get a better understanding of the social and biological history of diseases in ancient Rome. I found it super cool, and I was honestly looking forward to this workshop. And just a few reminders for the attendees. Jessica has 2 links in the chat for everyone. Yeah, Binjo like provided it for us in there, so you just have to like scroll up like a little bit. And she also provided the link to our 2 next events, which are the Healthcare Careers Panel and our closing keynote speech by Dr. John Bodish. So I think you guys should check those out. It's really cool. So thank you again for attending this workshop, and we look forward to seeing you in our next 2 events.
Thanks so much, everyone.
That was great, Jessica. I really love that.
I'm so glad. Sorry, it was so rushed. I was like, oh no.
Oh, it's okay. It was actually good. I feel bad for having to cut you off. Oh my gosh.
No, not at all. I thought I had more time, then I looked down at my clock and was like, woo, okay.
Okay. Yeah, thank you again. I'm just gonna have to like end this meeting now to pass it on to the next person. Thank you. That was amazing.
Bye.
Bye.